Hi everyone, my name is Chase Minardi. Today's date is September 9th of 2025. Here's my paper to show it, right? And this is my week 3 cell bio project. Today, I'll show you a real-life example of a prokaryotic and eukaryotic interaction in the human gut. Amonophilia signals to the intestinal epithelial cells through its protein channel AMAC 1100. So let's meet the players. On the left is a human intestinal epithelial cell. It's about 10 to 20 micrometers tall, contains a nucleus, mitochondria, endoplasmic reticulum, Golgi apparatus, tight junctions, and apical microvilli. On the right is your Akkermansia mentophilia. Sorry if I'm butchering that. It is a bacterium roughly 0.6 by 1.5 micrometers that lives in the mucus layer of your gut. Notice the scale bars. The eukaryotic cell is an order of a magnitude larger, where the prokaryote's not. Eukaryotic cell also contains organelles, where the prokaryotic cell does not. So, where do they meet? They meet in the epithelial cells forming a barrier-sealed tight junction covered by the MuC2 rich mucus. The Akkermansia sits in this mucus and presents an outer membrane protein previously mentioned, the ACMU 1100. The host cell will then express TLR2, a pattern recognition receptor. When they bind, it starts a signaling cascade inside the epithelial cell. Once downstream of the pathways, it will increase transcription of barrier-supporting genes. They have many results. The first one is the production of tight junction proteins, which overall tighten the pericellular seal between the two cells. MuC2 secretion is increased, the thickening mucus layer that separates the microbes from the epithelium. And finally, an increase in H5T biosynthesis, which is serotonin. Nearby cells will produce more serotonin, improving gut motility. So, this is evidence supported. A 2021 primary study shows that it increases intestinal and circulating serotonin, improving GI motility, especially in antibiotic-treated mice. Crucially, the TLR2 signaling pathway is required. A similar 2024 review summarizes a broader ACME 1100 effect, as well as clinical interest in the postbiotic, where there is a defined therapeutic potential with these interactions. This is important because the bacterium thrives in our host mucus. The host gains a tight barrier and improved physiological tone. Such specific microbe-host molecular dialogues help explain the microbiome shakes, immunity, barrier function, and even neuromodulation. So at the end of the day, a single bacterial surface protein, ACME 1000, can talk to a human epithelial cell on the TLR2 pathway and change the host cell behavior. Tighter junctions, examples of a prokaryotic-eukaryotic interaction at the cellular and molecular scales. Thank you for watching. References are listed at the end of my written description. Questions are welcome.